Now that we've looked at cell anatomy, or all the structures of the cell, we can look at cell physiology, or how the cell operates. In this particular segment, you need to be able to list and describe 3 passive and 2 active transport mechanisms used by the cell. List and briefly describe the 2 major periods of the cell cycle. List and briefly describe the events of mitosis. List and briefly describe the 2 steps of protein synthesis. Describe the role DNA and RNA play in protein synthesis. And list the 3 types of RNA and briefly describe the role they play in protein synthesis. As we begin to look at cell physiology, you have to remember that cells are the smallest living thing. As such, they have to carry out all of the life processes. And you'll remember there's a long list of them. 3 of them that we're going to examine as we look at cell physiology will be membrane transport. Now, the cell membrane acts as a barrier between the inside and the outside of the cell. So this is that part of life process about maintaining barriers. We're going to look at protein synthesis, which is how the cell uses some of its energy to make something it needs, a protein. So that's metabolism. And we're going to look at cell division, which is how the single cell divides. So this is its form of reproduction. Let's start by looking at membrane transport. You'll remember that one of the basic principles of anatomy and physiology is the principle of complementarity. That is, things have their particular structure because of how they function. Well, the cell membrane is that phospholipid bilayer. You'll remember that there are all of those phospholipids with their little water-loving heads, their phosphate heads pointing out, and their little lipid tails pointing in. And there's some proteins that are embedded on it, in it, go all the way through it. Some cholesterol here. And it is the function of the membrane to maintain a barrier between the inside and the outside of the cell. Now, this particular barrier is selectively permeable. Permeability means things can go through it, and selective means it doesn't let just everything in. So that the cell membrane controls what enters and leaves the cell. Your cells are bathed in interstitial fluid or extracellular fluid. Basically, this is a solution of nutrients that are outside the cell. Now, a healthy cell is going to need to move nutrients from outside themselves into themselves. So we're going to have to move material from the interstitial fluid into the cell. Intracellular fluid is that solution inside the cell. If a cell has metabolic activities going on, there are going to be some metabolic waste products. If these are allowed to build up into the cell, they will damage it and kill it. So we have to have a way to get these waste products out. The cell membrane, because of its selective permeability or its differential permeability, allows some things in and some things out. That's the whole function of the cell membrane. Now, cell membrane transport mechanisms can be passive in that there is no cellular energy expended. These kinds of transport mechanisms occur because of the laws of nature. And this would be diffusion. And there are 3 types of diffusion. There's simple diffusion, facilitated diffusion, and osmosis. And then we would have filtration. The other type of membrane transport mechanisms are active. Now, in this case, the cell uses ATP to move material across the cell membrane. We'll look at solute pumps, and we will look at vesicular transport as the active processes used by the cell. Diffusion is the molecular movement across a membrane along a concentration gradient. Now, the membrane has to be permeable to the molecule that we're moving. That is, there has to be some way for the molecule to cross the membrane. And when we say along a concentration gradient, we mean that we're moving the molecule from an area where the molecule is highly concentrated into an area where it's not as concentrated. Diffusion is going to act based on size of the molecule and solubility of the molecule. Now, if we look at diffusion without a cell membrane involved, Diffusion works and does not require any energy on the part of the cell because molecules and substances are in constant motion. In gases, they're moving quite a bit, in liquids, not quite so much, and in solids, they're moving very little, but they are moving. As a result, molecules bump into each other from time to time. We call this molecular movement kinetic energy, and that's the driving force behind diffusion. Now, if you were, and you can do this at home if you want to check it out, if you were to take a lump of sugar and just put it in a cup of water, just drop it in there, and leave it long enough, eventually it would totally disappear because the molecular movement of the little sugar molecules would start to bump some of them off into the water, and that would expose some more to be bumped off in the water, and then them bumping into each other in the water, they would eventually evenly distribute themselves in the water. How fast this happens will depend on the size of the molecules. Small molecules are going to move faster than large molecules, and on the temperature. In the cold, molecular movement is very slow and sluggish. In the heat, it's much faster. So if you were to conduct this experiment with a glass of cold water and a glass of hot water, you would see the two disappear at different rates. The hot water would move much more quickly, would dissolve much more quickly. Whenever we have this uniform distribution of molecules, we say that diffusion is in equilibrium. Now, there is one thing about Mother Nature. Mother Nature hates any form of inequality. She tends to want to equalize things out. And this is true of the cell membrane. Whatever is outside the cell membrane, she wants an equal number inside the cell membrane as well, so that all of these membrane transport processes are aimed at achieving equilibrium. Now, simple diffusion occurs whenever you're dealing with nonpolar or lipid-soluble molecules. Lipids are nonpolar themselves, and Mother Nature has a real simple rule about solubility. Like dissolves like. So nonpolar substances, like lipids, dissolve each other, and polar substances, like water, dissolve each other. So things that are polar dissolve readily in water. Things that are nonpolar typically dissolve in lipids. And you'll remember our cell membrane is mostly lipid. So that things come up to the membrane. They simply kind of dissolve in the membrane and just dissolve themselves through. We're still going to move along a concentration gradient. There's going to be more of the molecule on one side of the membrane than on the other, and they will move through until they reach equilibrium. Now, some things that pass across the cell membrane by simple diffusion are things like our respiratory gases, oxygen and carbon dioxide. They are lipid soluble. Your fat-soluble vitamins, vitamin A, D, E, and K, readily just pass right through the cell membrane. Steroid compounds, steroids, remember, are lipids, so they pass right through the cell membrane. And alcohol. Alcohol will pass directly through the cell membrane. Now, this is a non-specific process. All it is based on is size of the molecule and solubility of the molecule. Obviously, we want things like oxygen and carbon dioxide to get into our cells and out of our cells if necessary, and fat-soluble vitamins to get in. Alcohol, though, is damaging to our cells, so if we had a choice, we would probably choose not to let this one through. But because of its solubility and its size, it does enter our cell membranes. By the way, this ability of alcohol to cross your cell membrane so readily is why you can get drunk so fast if you drink on an empty stomach. If there's no food in your stomach to absorb the alcohol, it just goes right into your bloodstream. If there is some food, that kind of sops up the alcohol and slows down the process a little bit. In facilitated diffusion, there is an additional component. There is one of these protein carriers or a protein channel in the cell membrane. So some of those proteins that are embedded in the cell membrane act to assist with facilitated diffusion. Now, we still move along a concentration gradient. There's left to be more on one side of the membrane than on the other, and this will occur until we have equal numbers of the molecules on both sides of the membrane. But this one, because proteins are involved, there's some recognition of the molecule. You'll remember that proteins are very specifically shaped, and that allows distinct proteins to recognize very specific molecules, so that a protein carrier will pick up only one type of molecule and allow it through. Not anything else will get through. So here we have some specificity. Things that tend to move in and out of the cell by facilitated diffusion are things like glucose and amino acids, which of course are our nutrients, and ions. Ions, even though they're very small, are charged particles, and they typically have to have some sort of doorway in the cell membrane to let them in. This means that facilitated diffusion does have some selectivity. There has to be a protein that recognizes the molecule, or there has to be a pore that's just the right size to let the molecule through. The third form of diffusion is osmosis. And osmosis is the diffusion of water. Water is our solvent for all of the things that all of our nutrient soup that we have, so that water is our solvent in living things. Now, water easily passes back and forth across the membrane, but water is polar, and we said the lipids were nonpolar, so they shouldn't dissolve through. So we believe that there are protein channels called aquaporins that allow water molecules to just go back and forth readily. Either that, or there are some tiny spaces between the lipid tails that allow water to move back and forth. So, in osmosis, water moves across the membrane, and this happens when the solute particles can't diffuse. In other words, the membrane is impermeable to what's dissolved in the water, but there's some sort of inequality across the membrane. Mother Nature doesn't like that, remember, so she moves water. Now, because she's not moving a solute across, or we can't equalize the same kind of molecule, but Mother Nature wants to make sure that the concentration of molecules on either side are the same. So they may not be the same molecules, but they're there in the same concentration, so that each side of the membrane is a 1% solution or a 3% solution of whatever we're talking about. As a general rule, your charged particles, your ions, do not diffuse readily across the membrane, so that whenever there's an imbalance in ions across the cell membrane, osmosis tends to play a role in equalizing those rather than diffusion. We talk about osmotic pressure. Whenever one side of the cell membrane is more concentrated than the other, we either move the molecule using facilitated or simple diffusion, or if the molecule can't move, we use water. And when water is moving, we talk about the pull that one solution has for another, across over the other for water, so that the pull of water across the membrane is the osmotic pressure. Now, in interstitial fluid, we have to have the same number of molecule particles, or particles in solution, as we have in the intracellular fluid. That's part of Mother Nature's inequality. Now, it turns out that a 9/10% solution of sodium chloride has the appropriate number of particles. This does not mean that the solution inside the cell is 9/10% sodium chloride, or the solution outside the cell is 9/10% sodium chloride, just that that's the same number of particles. So water is going to move across the cell membrane to help maintain those numbers of particles in equality on both sides of the membrane. When water moves into or out of the cell, it changes the shape of the cell, or the tone of the cell. Now, when you think of tone, think of muscle tone. A nice firm muscle is well-toned, and a flabby muscle is not so well-toned. And that's the kind of tone we're talking about, how tight and full something is. So osmosis has to do with tonicity of the cell, how much water is inside the cell. And with that in mind, there are then three different types of solutions we can talk about. In an isotonic solution, iso means same. So it's the same tone. The concentration of the solute is the same both inside and outside the cell. It's not necessarily the same stuff, but the concentration is the same. So since we said that 9/10% sodium chloride was the equivalent of what's inside most cells, we'll have a beaker of 9/10% sodium chloride, and we'll drop a cell into it. Now the cell, the tonicity, or the concentration inside the cell, is equivalent to 9/10% sodium chloride. First of all, is Mother Nature unhappy about this? No, it's the same inside the cell as outside the cell. No big deal. Water moves freely back and forth across the cell membrane, but for every water molecule that moves out, one moves in. So while water is always moving back and forth, there's no net movement of water. There's no big movement of water in one direction or the other. So the cell stays the same shape and size it was when it was dropped in that solution. The second type of solution is the hypotonic solution. Now hypo means less than or under. Think about a hypodermic needle goes under the skin. So in a hypotonic solution, the solution is less concentrated than the cytoplasm of the cell. So this time we'll put 5/10% sodium chloride in our beaker. The inside of our cell is still a 9/10% sodium chloride equivalent solution. Is Mother Nature unhappy? Yeah, it's not equal on both sides of that membrane. We got problems. And if sodium and chloride would diffuse, Mother Nature would take care of this problem by having the sodium and the chloride move across the cell membrane until we reached equilibrium. But sodium and chloride are charged particles, and remember, they don't move across the membrane. So we can't use diffusion. We're going to have to use osmosis, which means water is going to have to move. Now here's the question. Which way does water move? Into the cell or out of the cell? Now, before you think about that, before you get too confused, water has got to dilute the more concentrated solution. So which is more concentrated, 5/10% or 9/10%? I hope your math skills are sharp enough that you picked 9/10%, because what's going to happen is water will move from the solution in the beaker into the cell to dilute down that until we reach the same concentration. Now, as the cell takes on water, it's going to swell up, and if it takes on enough water, it will rupture. We call that lysis when the cell does that. Our third type of solution is the hypertonic solution. Here, the solution is more concentrated than the cytoplasm. Everyone knows what a hyperactive child is like. Well, hyper means more than. So this time we'll put a 2% solution of sodium chloride in our beaker and drop our cell in. And the interior of the cell is still equivalent to a 9/10% sodium chloride solution. Mother Nature is again unhappy because we do not have the same percentage on the inside and the outside of the cell. But diffusion can't help us because sodium and chloride won't diffuse. The membrane's impermeable to them. So once again, we're going to have to pull out some water, use some water to change this up. Again, which direction is the water moving? Into the cell or out of? Remember, we have to dilute the more concentrated solution. So water's got to move out of the cell into the solution in the beaker. When this happens, the cell shrivels up. And it's hard for us to realize that the cell is dehydrating because it's sitting in there in this watery solution. How can it be dehydrating? But it is. It's losing water, and that's what dehydration is. Now, these are red blood cells. Red blood cells don't have a nucleus, and they have normally this little biconcave shape with this little dimple in them. This is a red blood cell in an isotonic solution. There's no change. It stays the correct shape and size. If we put that red blood cell into a hypertonic solution, remember the hypertonic, the solution is going to be more concentrated. We're going to pull water out of the cell into the surrounding solution, and the cell is going to shrink up. Now, when this is a blood cell, the particular word we use is crenation, but it's sort of a special word for blood cells. And here we have the same blood cell in a hypotonic solution. Remember, here the solution is more dilute than the inside of the cell, so water moves into the cell to dilute the concentration of the cytoplasm. There you see the cell swelling up, and if it takes on enough water, it will rupture or lyse. So, usually when you're given IV solutions, they are physiological saline. That is, they are 9/10% sodium chloride. Why do you think that's the standard IV solution? If we give you something in your body, we don't want to damage your cells. Physiological saline is going to have the same tonicity as your body fluids, so your cells will not be damaged by this solution. Sometimes hypertonic solutions are given to relieve edema. Now, edema is extra fluid in your tissues, and if we put a more concentrated solution in your blood, fluid tends to leave the cells of your tissues and enter the bloodstream where it can be eliminated through the kidneys. So hypertonic solutions can help relieve edema. On rare occasions, they may give hypotonic solutions to relieve severe dehydration. Now, with severe dehydration, you've been without water for a long time. Your cells are all just shriveled up, and we want to bathe them in a hypotonic solution so that they'll take up water readily. Now, this is dangerous to do because you can overdo it if you're using it through IV. Typically, they try to rehydrate people with oral use of water rather than administering a hypotonic solution, but they have been known to do it in severe cases. The other passive process is filtration. Now, diffusion was driven by kinetic energy, molecular movement. Filtration is driven by hydrostatic pressure or water pressure. We're going to move materials along the pressure gradient. There'll be more pressure on one side of the membrane than on the other, and we'll push molecules through because of that pressure, so that the solvent is forced through pores or tiny little holes in the cell membrane. The size of the hole in the membrane is going to limit which molecules or particles get through. So this is a fairly non-selective process based only on molecule size. Basically, you can't squash a basketball through a hole that's only big enough for a softball, so there'll be certain limits to which ones can get through. Filtration is used when we mix cerebrospinal fluid, but we'll also talk about it quite a bit when we talk about urine formation a little later on. The active transport processes require the use of ATP. The first process we'll look at are the solute pumps. Now, active transport is molecular movement against a concentration gradient with the use of ATP and the use of a carrier protein. We're going against the concentration gradient. This time we're going from where there's a lower concentration of the molecule to where there's a higher concentration. So we're pushing uphill, and that's going to require energy to do that. And if we're going to spend some of that energy, we want to make sure we get what we're buying. So we use a carrier protein because carrier proteins have that recognition feature. What happens is the carrier protein recognizes a particular molecule and attaches to it. And then the ATP changes the shape of the carrier protein, sort of like opening a door, and pushes the molecule across the membrane. Here's an example, what we call the sodium-potassium pump. Here the protein carrier is open and sodium is recognized and attaches to its little seats inside the carrier. If ATP is present, as the ATP breaks down, it adds its high energy phosphate to the protein, and it's literally like a swinging door. The door closes on this side and opens on the other side, and the sodium molecules are pushed out. We also open up a couple of seats for potassium ions, so the potassium joins in, and just the fact that that energy burst is momentary, doesn't last long at all, as soon as the energy is over, the door swings back to the other way, because this is its natural way to be. But it pushes the potassium ions across this way. So in order to pump sodium and potassium across the membrane, we have to use this energy. So far we've talked about molecules or ions, but what happens if we have sort of a large particle, something bigger than a molecule? That's when we have to use vesicular transport, sometimes referred to as bulk transport. And the processes here are endocytosis, which is moving something from the outside of the cell to the inside, or exocytosis, which is the reverse of that, moving something from the inside to the outside. Now, one type of endocytosis is phagocytosis. This is what we refer to as cell eating. Basically, some large particle, this is bigger than a molecule, is taken in by the cell. There is a series of reactions that occur that cause the cell membrane to sort of move together. Some pseudopods come out and surround the material. These little purple things are some of the proteins that are embedded in the cell membrane, and they're part of that process that helps determine where we're going to do this. The cell membrane is really neat, that fluid mosaic model. Whenever the cell membrane touches, it just bleeds together. So it just kind of seals itself up and pinches off a particle to the inside. And so we'll have one of those vesicles that we talked about in cell structure. This one's going to be called a phagosome because it was created as the result of phagocytosis. And the particle is going to be inside the cell, surrounded by a little bit of cell membrane. Pinocytosis is another form of endocytosis that's referred to as cell drinking. Sometimes the material we bring in is more of a liquid nature, like droplets of oil perhaps. It's the same process of sending pseudopods around some of this material. The cell membrane touches, we pinch off the little vesicle, and the little oil droplets are now inside the cell, inside this vesicle, instead of being outside the cell. And receptor-mediated endocytosis uses some of these proteins as receptors. So they're very specific for a molecule. When the receptors get full of some molecule, we start that pinching off process. We get a vesicle inside the cell with all of the little molecules taken in. And once these molecules have been used up, the vesicle can return back to the cell membrane. These little proteins can return back to the cell membrane and be available to pick up more particles. Receptor-mediated endocytosis is a good way for the cell to regulate how much of a particular molecule it takes in. Iron, for example, can be toxic to the cell, but cells that need iron have to take in just what they need. By having only a certain number of receptors for iron on the cell surface, when those receptors are full, all of these little receptors for iron will be inside the cell, but once the iron comes off of the receptor, it can be recycled back to the surface so the cell can take in more iron. This way, only the amount of iron the cell needs is taken into the cell, and the cell doesn't poison itself. All right. When a white cell ingests a bacterial cell, it uses which process? Exocytosis, phagocytosis, receptor-mediated endocytosis, or pinocytosis. Now, a bacterial cell is a pretty large molecule, not a molecule, a pretty large structure. Phagocytosis is the correct answer. We're eating something, we're taking in a big thing. Exocytosis would be the cell getting rid of something. Receptor-mediated endocytosis would require that we have a receptor, and pinocytosis would be taking in some sort of liquid. The cell membrane is permeable to glucose, and cells use up glucose pretty much as fast as it enters the cell because that's how we make our ATP. If a cell is placed in a 5% glucose solution, what's going to happen? Will water move into the cell by osmosis? Will water leave the cell by osmosis? Will glucose diffuse into the cell? Or will glucose diffuse out of the cell? Glucose diffuses into the cell. We know diffusion is going to be the process because the membrane is permeable to glucose, so diffusion will operate. If the membrane were impermeable, then we'd have osmosis going on. Because we've got 5% glucose outside and pretty much no glucose inside the cell, That's going to be our concentration gradient from the outside to the inside. Another process of the cell is cell division. This is the reproductive process. And cells have a life cycle like everything else. That's basically two major parts. We have interphase, and then we have cell division. Now, interphase is all of this metabolic phase. The cell is being a cell, it's doing what it needs to be a cell. But during the part of interphase, we get ready for cell division. That is, we synthesize DNA. Get it ready so we have another copy. And then cell division is the actual division of the cell itself. Let's talk a little bit about DNA replication. This means we need to make an exact copy of all the DNA that's in the cell. The DNA is the blueprint for the cell. The cell can only do what it's supposed to do if it has the same DNA as every other cell that it came from. So we have to have a mechanism for copying that blueprint and faithfully passing it on to every cell that's manufactured. You'll remember that DNA is a chain of nucleotides. It's actually a double chain, and it kind of folds up into a spiral staircase sort of thing. We call it a double helix. And the two chains of nucleotides are held together in the center by hydrogen bonds, which are fairly strong attractions but not really strong bonds. So that we have our double-stranded DNA molecule, and these little dotted lines indicate the hydrogen bonds. And your nucleotides contain either adenine, thymine, cytosine, or guanine. So the first thing that has to happen is the double strand has to unzip. We have to break those hydrogen bonds and unzip it. Then we have to use each half of the molecule as a template. That is, this is half the molecule. If we could build the other half faithfully and use this to build the other half, we'd end up with two strands that were identical to the first strand. And the way this happens is through something called precise base pairing. Now, these little nucleotide pieces are floating around in the nucleus, the nucleoplasm. They're just there. They're part of what the cell keeps manufactured, so that it's sort of like we've got the building blocks. We just have to make sure we put them in the right order. And precise base pairing makes sure that this happens. Now, what this means is adenine always pairs with thymine. So when you have a naked adenine, we're going to look for a thymine. There's an adenine, and see why we found a thymine to go there. And cytosine always pairs with guanine. So whenever we have a cytosine here, the building block that's going to automatically go up there is guanine. So we have our two copies of DNA. That's critical. Now, in this interphase part, this first part is the variable part of the cell cycle. This is the cell just doing its thing. There is a point right about here where there is a buildup of some sort of signal inside the cell. Scientists have been looking for this signal for a long time, because if they could find this signal, they could trick cells into dividing faster. But whenever it hits that signal point, at that point on, the rest of the cell cycle, the rest of interphase and this cell division phase, is going to occur pretty rapidly in pretty much a standard amount of time. Because once we get to this trigger point, that's the trigger to make our DNA. That's what happens here. And then in this last stage, we make a little more cytoplasm. We replicate the organelles so that we have plenty of those to go around, and we're ready to go. When we start the cell division phase, we've done all of our prep work. We now have to divide the nucleus, which is mitosis, and then divide the cytoplasm, which is cytokinesis. And you'll notice they overlap a little bit. We've got mitosis, the nucleus division process going on, and then we start dividing the cytoplasm and finish that up, and that's how we finish up. And we end up with two cells that are identical to the original cell. And both of those cells start into an interphase until they reach their critical point, and then they finish up with replication, some more growth, and then they go through the mitosis phase. Now, mitosis, the nuclear division part, can be divided into 3 stages: prophase, metaphase, anaphase, and telophase. In prophase, the nuclear structure begins to disappear. That is, here you sort of see the nuclear membrane starting to break down. The chromosomes coil up pretty tightly, so they start becoming visible. Normally in the cell, they're kind of stretched out, but we don't want to get them too tangled up during this division process, so we coil them up pretty tightly, and that's when we start seeing the strands of chromatin here. Now, because we've made an extra copy of everything, in order to make sure that we divide those correctly, that is, we send one copy to one cell and one copy to the other cell, we hold the replicated chromosomes, the duplicated chromosomes, together with a structure called a centromere, so that this keeps the 2 duplicates held together until it's time to send them to their respective new cells. Those centrioles typically are not that visible, but they become more visible, and you'll remember that the function of the centrioles was to make microtubules, and spindle fibers are special microtubules that are involved in cell division. And in this particular view, we don't really see it's a little early to see the centrioles and the spindle fibers, but they're just getting ready to start. The second stage of mitosis is metaphase. Now in metaphase, all of those chromatids line up on the equator of the cell. And here you can see the centrioles a little more clearly, and these little strands are the spindle fibers. Now a spindle fiber from each side has attached to one of those duplicated chromosomes, one of those chromatid pairs. So that when we go into anaphase, the next phase, what happens is the centromeres that were holding the chromosome pairs together divide, and those spindle fibers start to shorten up, and as they begin to shorten up, they pull the chromosomes to opposite ends of the cell. So here's one set of chromosomes, and here's the other set of chromosomes. Now there are a few spindle fibers that actually get longer. They're not attached to chromosomes because they're going to push the cell into more of an oblong for the cytoplasm division process. And then the final phase of nuclear division is telophase, and we begin to have the spindle fibers go away, the centrioles disappear, the nucleus starts to reform, and we begin to see strong indications that cytokinesis is occurring. The cell membrane is pinching in on itself. We call this a cleavage furrow, and the 2 pieces of cell membrane will get closer and closer and closer together, and you'll remember when cell membranes touch, they just kind of heal each other off, and so we will have 2 new cells, both with the same DNA, both identical to the original cell. The third thing we're going to talk about is protein synthesis. We've mentioned that the DNA is the blueprint for the cell, and the reason it is because the DNA controls all of the proteins that are manufactured by the cell, and you actually are a result of all the proteins your cells can make. Every single metabolic process that occurs in your cells has to be mediated by an enzyme, and an enzyme is a protein. So every protein you make has an effect on every piece of your cell's life cycle. DNA is the basic blueprint for all of the proteins your cells can make. Genes are simply segments of DNA that code for one protein. And as I said, the proteins determine the cell function because they manufacture the enzymes and all of your cellular structures. Because DNA is the master blueprint, it's in the nucleus. And typically, we do not take the entire master blueprint to the building site, but only the piece that we're interested in at any given time. So in order to protect the DNA, we copy the DNA to an RNA molecule, and the RNA molecule carries the information to the building site. For a long time, it was hard to figure out exactly how the DNA carried the genetic code. But they finally stumbled upon the fact that the nucleotides on the DNA would code for the proteins. Now we've got 20 different amino acids that we have to string together to make our proteins. So it's kind of like, okay, how many base pairs does it take to give us 20 different unique codes? And it turns out that if you have 3 nucleotides, you can code for 64, you have 64 unique different code words. Now that's more than we need, which is a good thing, but that's how they determined that 3 nucleotides made one of these genetic words. They coded for 1 amino acid. So on the DNA, this 3 nucleotide sequence is called a triplet, but on the RNA, it's called a codon. Now they figured out the genetic code by looking at RNA. So when we look at the genetic code, it's always RNA that we're looking at. And we know it's RNA because RNA differs from DNA in one couple of features, but one important one is the fact that RNA does not have thymine, but it rather has uracil. So when you see these U's, you know you're dealing with RNA. Now notice that because we've got more code words than we need, we have a lot of these amino acids that have 2 or more codons that will code for them. This means that Mother Nature will allow you to misspell the genetic code slightly and you'll still get the same information. There is one codon down here that is the start codon. It also codes for the amino acid methionine, but it's the start codon. This is how the RNA or the ribosome knows where to start. There are 3 codons that are stop codons, so that the ribosome knows that when it hits any of these 3, it's done. It can release and it's ready to go on and do something else. The protein synthesis process is 2 steps: transcription and translation. Now when you transcribe something, you make a copy, and that's what transcription is. We're copying the DNA to one form of RNA, and this is called messenger RNA because it takes the message out to the cytoplasm. When you translate something, you convert it from one language to another. Translation is the process of using that code on the messenger RNA and turning it into a protein. Since RNA is a nucleotide and proteins are amino acids, we're basically taking a sentence written in nucleotides and turning it into a sentence that's written in amino acids. The transcription part occurs in the nucleus. That's where the DNA is. And a piece of DNA, not the whole DNA strand, but a piece of it, the piece that has the gene we're interested in, unzips, and using that same precise base pairing thing going on again, we make an RNA strand against the unzipped DNA. And it's the same thing, except that adenine on the DNA uses uracil when you're making RNA. Thymine still pairs with adenine, guanine and cytosine still pair. But any place where you have an adenine, if you're making DNA, you'd put a thymine there. If you make an RNA, you put a uracil there. Now, the messenger RNA is a complete transcript of whatever was on the DNA. And so some of our DNA, you may have heard, we've got some genetic junk. We have some stuff in our DNA that doesn't code for anything. We're not sure why it's there, but if Mother Nature has it there, it must have had a purpose at one time or still has a purpose that we don't know about. So we sort of make our straight copy off of the DNA as kind of like a rough draft. And we have to edit the messenger RNA, cutting out the parts that aren't important, and then we allow the messenger RNA to go through one of those nuclear pores, remember those are pretty good size, so it can get into the cytoplasm. And it's in the cytoplasm that we begin the translation process. You'll remember that the site of protein synthesis is the ribosome. So the ribosome has to attach to the messenger RNA. The ribosome actually exists as 2 subunits, a large one and a small one. And part of what they're composed of is ribosomal RNA. And ribosomal RNA helps pull the 2 subunits together around the messenger RNA and helps find that starting place so that we read in the right direction. The ribosome then reads the messenger RNA, And in order to bring the amino acids to the ribosome, we have a third type of RNA called transfer RNA. And transfer RNA brings the amino acids to the ribosome so that they can be strung together in the correct order so that we get exactly the protein we want. So out in the cytoplasm of the cell, because this is where translation occurs, we have this strand of messenger RNA that's been sent out. And our ribosome finds the start end and attaches the large and the small piece to it. Now transfer RNA looks kind of like this weird bent coat hanger thing here. It has these three little nucleotides right here. This is called the anticodon. And an enzyme reads that anticodon and attaches the appropriate amino acid to it so that a particular transfer RNA with a particular anticodon is always carrying the same amino acid. Now the transfer RNA comes over, it looks for its codon, the anticodon attaches. We get another one in here when we have the two amino acids side by side. Part of what the ribosome does is stick the two amino acids together. Once an amino acid is stuck to an amino acid chain, the transfer RNA is unloaded, it's empty, and it's released from the ribosome, and it can go out and pick up its appropriate amino acid again so it can be reused. So this is kind of like the trucking company bringing in the raw supplies, but they always have to go just to a particular place to dump their supplies. So just to review a little bit, transcription occurs in the nucleus. We make messenger RNA against the DNA. The messenger RNA, once it's edited, leaves the nucleus through a nuclear pore so it can be translated. The ribosomal RNA helps the ribosome attach to the appropriate spot on the messenger RNA. And the transfer RNA brings the correct amino acids to the ribosome, and using precise base pairing again, codon to anticodon, we put the right amino acid in the right spot. Once the amino acid has been attached to the long chain, the empty transfer RNA is released so it can go out and pick up amino acids again, and a fresh one comes in to the next reading spot. Once its amino acid is attached, we just keep moving down. So it's a process of these three reading sites on the ribosome. One is holding the whole chain because it's still got its amino acid attached to it. One is just dumped out the empty transfer RNA. The other one is waiting for the correct transfer RNA to come bring its amino acid. And we just keep moving down, unloading, sending off for a new one, bringing a new one in. Unloading, sending it off, bringing a new one in. So in summary, the cell membrane is semi-permeable, so it controls what enters and leaves the cell. Membrane transport is both passive and active. Passive processes do not require any energy on the part of the cell. Active processes do require energy, and the only cell energy available is ATP. Cells reproduce by mitosis, which involves dividing the nucleus and the cytoplasm. There are two major life cycle phases. There's interphase and then there's the cell division. Protein synthesis uses transcription and translation. Okay. During protein synthesis, amino acids are brought to the ribosome by which one here? Messenger RNA, transfer RNA, ribosomal RNA, or DNA. Remember, transfer RNA trucks the amino acids in. In the translation step of protein synthesis, all of the following are involved except, now this is translation, remember, messenger RNA, transfer RNA, ribosomal RNA, or DNA. Which one is not involved in translation? And that's going to be the DNA. DNA is involved in transcription, but once we've transcribed it, RNA is the only thing that's involved in the translation.